Hello! Welcome to the Microbiology Journal Club. I'm Fazlam. This is my colleague Danny Cham, where we talk about all things great that are small. Um,
Yep. Yeah, we've been doing this for a while now. This is our on week, if you will, where we dive into a paper. We go figure by figure and explore what the data tells us. Basically, read the story. And on the other weeks, we choose the paper um, and cover some news headlines.
yeah, and this week we've got a really interesting one that delves into how coronavirus makes more of itself, how it manipulates the cell into making more of itself, and even more so,
Yeah,
like
and
the interesting technologies that allow us to see it.
absolutely. Yeah, I mean, we haven't talked about any structural biology like
Yeah.
this. Yeah, I don't think we.
Yeah,
Yeah, we.
we tiptoed into it with a few like other papers looking into spike protein. We look into a bit of
Mhm.
like structures, but those papers often use crystallography, which
Right.
is
Well, I think those papers, like the papers that we have talked about that had the spike, they already had the structure. It's
right.
not like they determined the
Yeah,
structure. They had the structure, and then they looked at epitope mapping, and they said, okay, here is on the 3D structure where that antibody would sit.
yes,
Um, but this is one of those primary data collection papers where they're actually figuring out like what what the shape of something is.
yeah. I know. This is a technique that's really quite new, and that
Hmm.
and yeah,
A revolution, really. I
yeah,
mean, uh, called cryo EM, right? It's kind of interesting because when we chose this paper, I had heard of cryo EM and I knew that it was helping to bridge the gap in sequence to structure, right? People always say we're sequencing tons
yeah,
of things, but we don't know the structure of a whole bunch of things. And this technology in the past, I don't know, maybe I think 2013 it first came out,
yeah.
so like almost a 10 years now, like has been helping to bridge that gap. So like that's how I knew it. I'm like, oh, this is a way to find structures really easily, and it goes really and it's like faster than crystallography, as you
Yeah.
mentioned, which like takes a bit more time.
Oh,
but but this application is even more like stretching the limits because it's not single molecules they're looking at entire like chunks of cells.
yeah. They're
Yeah.
looking at the molecules in place as a so with
Hmm.
one of the criticisms of crystallography is that you just take a purified protein and just put it into chunks and chunks of it and then average that out to get the kind of structure. But that's not
Hmm.
natural. That's not seeing the protein as it is inside the cell. And I thought that was for a long time. I thought that was impossible because of the way that what light waves work and how
Well, people did the TEM is old,
yeah,
right? Like that's from like the 70s, maybe the 60s. Like that was, but they embed that in resin.
yeah,
So like there's like this, of course there's with any technique there's an artifact that might emerge, right? If you're treating the biological material, like when you take a slice of a tissue, you never see it like, wet flesh just sitting on the slide. It's like alcohol and like different agents to make sure that like things don't rot away and that you can see colors clearly and contrast. So TEM was like resin. They use like
oh
plastic
yeah, and they don't
basically.
even use light. They use electrons, which they can get a
Mhm.
much shorter wavelength, which means that they can get a lot more definition from cells. And I thought,
Yes. Oh, well, there's electrons here too,
yeah,
right?
this
This is
is
also electrons.
electron microscopy, but also they do it in a very smart way to get a high resolution on the tissues as well, uh,
Mhm.
because and you can get a lot of like you see a lot of intracellular stuff with electron microscopy, but to the level of actually seeing molecules is something I've never really seen before. Um,
Yeah. I do believe that computers help with that.
yeah, I,
I think there's because I mean they talk about it too. We'll get to it in one of the figures. They talk about like assuming symmetry.
yeah,
And like so that's something where I think on the computer side they're like, okay, we've seen enough examples. It probably looks like this, right? They average images
yeah.
together to get something. Yeah.
Yeah, and I think they do that somewhat with lots of different structures. It's the fact that they do it in a in its natural like state, in its natural conformation.
Yep.
That's the amazing
Absolutely.
thing for
Yeah.
me. So
Yeah, and I think it's important. Oh, I guess we can talk. We can go into see the first
yeah,
slide because we sort of do explain this technology. But I will say that it's based on this idea, like as I said, like resin is how they did TEM. The cryo part of the cryo EM, which makes it, I guess, a significant technique. Again, 10 years now, maybe longer, but is just like if you freeze ice fast enough, it doesn't make a crystal. So like that's right. That's this idea, like, when you freeze things, freezer burn or whatever, right? Like if you freeze your vegetables, they get all mushy when you soften them up, and like that is, ice crystals breaking the biological structures. But in vitrification, which is what they call it, the ice doesn't get to form those crystals. It just I guess they call it like a glass-like state.
Yeah.
The water just stops moving in whatever position it was in.
Yeah, which is, I mean, brilliant, because that's, as you said, this is the biggest problem, and also that means that everything's should be in the same conformation. So
Yes,
it should look like the cell should look without these giant ice crystals forming on it. And
Right. Right. Yeah, like things aren't going to get pushed around as like, we know ice expands, right?
Yeah.
So things aren't going to get pushed, like a crystal's not going to appear and then push away these membranes or like disturb like the molecular structures. But yeah, and so I guess this is an image, right, of the tech.
Yeah, this is, I mean, would you like to take me through this actually?
Yeah, sure. So they have a cell, that's A,
Yep.
right? And actually, that cell has already been milled, as they say. They use I mean, this is like some crazy physics, I guess, but there's a beam that has electrons that lets you see through things, right? Electrons substitute for light. Those are very tiny subatomic particles. Larger subatomic particles are like metal ions.
Yeah.
I think they use gallium is what I read. And they just shoot ions of metal, and that like blows away chunks of material. I guess it vaporizes it probably. Yeah. It's all happening under a vacuum, so the metal like doesn't hit anything in the air, right? Like air would interfere with that process, and they just with computer precision, they blast away layers of the cell until what's left is just a thin, like nanometer thickness chunk of the cell. Yeah. And then they, with that thin surface, that they can then shoot the electrons through it and get an image.
Yeah, that's, I mean, previously, like, for microscope slice, we want to get the thinnest slice possible.
Mm-hmm.
And previously they're using things like freezing the same way, but using like a diamond like razor to try and cut things to get things.
Yeah, diamond blades.
But even then, that doesn't get things quite thin enough. It's not precise because that can cause some like scratching and some artifacts to happen where even that blade isn't like perfect. Whereas this focused ion beam, you can almost get like that perfect thinness, and you can get things.
Well, I mean, it's interesting, right? Yeah. Because when you think about the physics of it, like when you think about a cutting surface, right? Like a blade is like some amount of atoms, right? Are like in like this like V shape that's gonna like dig into your other set of atoms and start pushing off, right? Part of it. But with a beam, or almost like a laser, right, if you will, it's not a laser, but you can imagine like that sort of style. Like it's just this. As tightly as you can focus it is as tightly as it can cut.
Yes.
Right. Like the space of that is a lot smaller than what you might have on like an edge of a blade.
Yeah, and they get so thin in this paper, because I think the nanometers, which is really, really tiny. I like to the point really like the wavelength of light is in that.
Yes, hundreds of nanometers.
Like, so, to the point where, like, I don't think you can even see these slices with light. They're that small, maybe, or...
Yeah, that's I wasn't even thinking about that. Yeah, it's not a concern, right? Because they're not even using light. And even when they show us the image of, like, actually, if you go to the next slide, this is like an image of, like, that cell. So basically, A, from this first figure, this is a real image of what A looks like. Yeah. It's like a little blob of icy cell that sits on a mesh, and then they blasted it away to make that thin little middle section. And they're not looking at this with light. They're looking at this with electrons. Yeah. Because, it's possible that light wouldn't even be able to capture what we're seeing here, right? Like, we need that small wavelength electron to be able to see this. So C is like a top-down image, right? That's where the electron beam would go down. but A and B, that's like the before and after they blasted it with ions.
Yeah. And once they do, they don't. They do like the kind of transverse, like taking the electron image. They also do this thing tomography where they take
Yes.
pictures from multiple angles as well, so that you can get
Hmm.
multiple angles on each molecule and then predict their 3D structure. Which
Hmm. Hmm. Yeah, I mean that's definitely something that I mean, yeah, you can understand that even from the light
yeah,
perspective, right? Like even in light, you can see tomographic things. Actually, do Apple photos do that these days, right? With the multiple cameras
I,
on the photo, and then you have that weird like shift you can kind of
I
see,
mean there are certain like
right?
types of 3D images where they tilt it and then you can see the background. I can't. Um,
Yeah, I know. I feel like Apple, I think some cameras do this these days on phones.
yeah,
They do like I mean, it's not a proper tomogram because they don't infer the 3D position of everything. But that effect where you see that from the same position, right? If you take the picture a little bit differently, you see a little bit more of different sections of the picture. And so the tomogram would like compile that information and make us like a 3D, a 3D
yeah.
image.
And I think that's also why it's quite important to have as thin slices as possible because then you get single molecules and you don't get because if you have too much in like in a certain like field of view,
Yes.
it will interfere and make it
Yeah.
you get a lot of noise
Yeah.
and background. So having it very thin means you can
Yeah.
look at individual molecules rather than seeing a bit of things in the background. I mean, it's
Yeah.
and
I mean, even though we're not looking with light, we still have that effect of
yeah,
shadows, right? If something's in front of something and the electron comes in and hits it, right? It's not a light particle that hits it and comes
yeah,
back, it's an electron. Yeah, you're not going to be able to see the thing behind it because, yeah. And so thinness is definitely a very important part.
yeah. So
Um,
this is really amazing.
yeah, so I mean, it is kind of crazy. If you go back to the first slide, like you were saying, tilting, right? That figure D has really shows you like that all this D and C, it's all in this little cartridge,
Yeah,
right? And that's being manipulated like with very precise gears, I
yeah,
suppose, like actuators, right? To make sure that they're just moving it ever so slightly in that electron beam. Yeah, and getting an image. So they do this with our favorite coronavirus. But also they also do it. I'm actually. It seems like most of the data from this paper comes from a different
yes.
coronavirus.
A mouse MHV. So mouse
Yeah.
murine hepatitis coronavirus, which is
Hmm.
uh, one where you don't need to like wear tons and tons of protective material to work with and sign off like
Yes.
and safety, going like, okay, right, I might die. Okay, it's not your fault if I die. I understand this is that sort of stuff that we've all had to sign at some point
Right.
when working with
Right.
these sorts of things. Um,
But previously they have done it on classic
yes.
SARS. There's plenty of literature in the classic SARS saying that when they do when they do TEM techniques, I don't know if they did tomography, but they did TEM techniques at least, and they saw like really interesting structures, membrane-bound structures where the virus seems to be replicating in. And they didn't exist in the infected form, but they exist in they don't exist in uninfected and they exist in infected. And when they use like you can even tag things in these images. Instead of using like fluorescence, you use like a chunk of gold,
Yeah.
a gold atom or something like this. So they're also able to see that, oh yeah, and there's like viral proteins that are attached to these things. It's a mix, right? Viruses use stuff from the host and
Yeah.
themselves, but they do make something that forces these, well, they call them organelles
Yeah.
actually, right? Replication organelles to come together.
Yeah, I think that will bring us to the first figure one, I guess, where we see these replications.
Yeah, at least,
Yeah,
yeah, let's look at a picture of what they did.
yeah. So they've got like a cryo EM tomography picture. I think that's what it's called. Let me check the figures. But yeah,
Oh
these
yeah.
are replication organelles that are created by coronaviruses when they affect cells, which are dedicated
Hmm.
to. So normal cells don't create these, but virus-infected cells suddenly create these big like blobs of that look like
Hmm.
just blobs of double membrane organelles.
Yeah. Well, it's interesting, right? So, we talked briefly about this. I don't think we ever like comprehensively reviewed, but receptor, right? There's a spike
Hmm.
protein that's the receptor for entry. It's an enveloped virus as well.
Yeah,
So even when when the virus makes contact with the outer membrane, there's some sort of endocytosis presumably that brings in that virus. So already there's a double membrane in that sense.
Yeah.
I don't think that's where this double membrane comes from, but like this is just like to give a sense of what's going on in the cell. So then you have a membrane that has the DNA or the RNA inside, and you have a membrane outside of that that's made of the host. And somehow that evolves into this formation,
Yeah.
right? Where I think it's two host-derived membranes now that encapsulate everything.
Yeah, they don't delve too much into how these are created. They do analyze their functionality, which
yeah. Well, you can see an image of one, or like one of the early stages, right? They have it's the one that has the virus particle in it,
yeah.
right? The viral particle envelope, right? So that structure is very much like as when the cell gets eaten up, right? Endocytosis.
Yes.
So there's the viral membrane, which was basically an old cell membrane from like the last time something got infected, and then you have this new host membrane around it. And things that go through endocytosis, they get put into this whole system of like vesicles and membrane-bound things that are floating around the cell, the endoplasmic reticulum, and also the Golgi. They're both like they're like a spectrum. When I was taught it in grad school, it's a spectrum of spaces, right?
Yeah.
That like have like a strong identity in one space and a strong identity in the other, and through like fusing of other little membrane bubbles, they sort of change and they can do different things. Right, so like they could digest something, they could transport something somewhere else. Right, these are like storage things in the cells. Shipping containers,
Yeah,
maybe is that a good analogy?
I think of them like as rooms where different things are going on, and
yes, but the rooms are like morphing,
yeah.
right? It's like holographic rooms.
Yeah, that's the problem. Analogies, like it's so difficult to find a good analogy for these. It's like they're rooms, but different people walk in and out. Yeah, so
they're multi-purpose rooms where like equipment can get shuttled between them, right, to change the function inside. Yeah, I like that.
Yeah, I mean, a bad analogy in the comments, and we'll rate it.
Yeah, for sure. And so the idea inside of an infection is the virus maybe makes something in that compartment, right, that starts changing the function of it. Like, oh, it's not going down the regular path anymore. That compartment is changing, and eventually it makes its way to become this double membrane vesicle, perhaps.
Yeah.
Right. It's not well worked. It's not within the scope of this paper to discuss it, and also it's still quite an open field of research.
Yeah.
In grad school, we shared the department with like hepatitis C virus professors. And I think in hepatitis C, there's a lot of research that goes on into this because, these structures have been identified, not just in coronaviruses, but in, other RNA-style viruses, positive strand RNA viruses, or even double-stranded RNA viruses. And the reason is that RNA in the cell is supposed to make proteins. If you have RNA in funny other structures, that's one of those innate immune signals that says something bad could be going on.
Yeah. So if your internal innate immune system to a cell sees double-stranded RNA, it goes, okay, time to put the self-destruct. And the virus is like, no, and
yeah, exactly. Yeah. So one of the ways to protect themselves is they can wrap themselves, right? So before they make too much of those danger molecules, which is their genomes basically, they make sure that they're protected from the cytosol. That's just a hypothesis. As to why these organelles exist.
It makes sense, but it's one of those things that does intrinsically make sense that it makes because I mean it's not like there are TLR like toll-like receptors are the things that would bind to these. If those were concentrated in those organelles, then we'd go, oh, that's crazy. But at the moment, that's our best guess.
Yeah. Yeah, at the moment they seem protected from the general cytosol, right? So they're made up of only what the virus wants to let in.
And we've got this this video of this as well that I can show quickly. So there's going to be a slight delay so I mean so we got this like tomographic image of it's just like looking at different layers of it, and then based on that, building off of that, they create a 3D model that looks really great. It's like spinning around and it's all cool, and then they show their favorite new like protein that they discovered at the end of it.
Yes. Yeah, you can see it. It's even in the earlier one, it was in red, right? They color coded everything.
So like in purple is the like ribosomes, and like and
Hmm.
in green is the DNA. The yellow is the all like the kind of double membrane
R,
network.
R, Green is the RNA,
RNA, yes, of
presumably.
course. I keep
But here's one of those things where like what they're seeing, there's no tags. They don't use any tags in all of
mm,
this. It's all just contrast or whatever, electron contrast. So what is something they can only really tell by shape?
yes.
And so what is the shape of DNA versus RNA? I mean, actually, they wouldn't be able to tell. It's just that it's thought to be RNA because it's an RNA virus.
Yeah, it's true. That is their best guess for things, and that's going to be. I mean, that is quite an interesting aspect of this paper because a lot of it is like they've got this brand new technology and they just point it at stuff, and then they go, "Look at this image.
Yes,
Look at this image. Look at."
absolutely, absolutely. What sort of clues can it give us? And they really squeeze the technique for everything it can tell us, right? Which again
Yeah.
is important because in other techniques, like maybe we can talk about this a bit later, but like traditionally maybe this could be done through some sort of biochemistry, right? Some sort of biochemical technique could give us some understanding of this. It could tell us the composition of all of these bubbles if we were
Yeah.
able to isolate them in some way. But that's very different information than seeing like where in the bubbles certain things happen, right? Or certain like concentrations are. So yeah, it's different information for
Yeah.
sure.
I mean,
It's just one
it's,
technique.
it's kind of convenient, I guess, because well, it's inconvenient because usually we want like all these lines of evidence, whereas a lot of the kind of things that we get here are like, here's a picture, here's what we think is going on, and
Yes, yes,
which is a
yes.
yeah,
Um,
it takes something
but
you
but
see.
again, the structure has been seen before in other
Yes.
viruses, so they have like a basis of seeing that before. And I didn't look into this whether or not in other viruses they have done some biochemistry to like purify out the things. They may
Uh,
have, but
yeah,
I don't
they do
actually
some
know.
biochemistry in this paper as well to like confirm like what's
Hmm.
in their new protein. Uh,
Right.
so shall we advance to the next?
Yeah, yeah, let's move on. So yeah, so they after doing this tomographic image, well, I guess they're gonna show us a whole bunch of different things they noticed,
Yeah.
right? In the
So we're
image,
going to be
yeah.
going through a lot of like slides that show bubbles with lots of grains in them that could be different structures, and also like I mean,
Yeah.
looking at the detail. So luckily, figure S1A is the one that's coming up now. So they've got these handy white arrows that point to all the kind of connections that are between
Yep.
different cells,
Yeah. Between different
between
organelle
different organelles.
bubbles.
Gosh,
Yeah.
yeah.
We should talk about what they call them double membrane vesicles,
Yeah,
right?
double membrane vesicles slash replication organelles. So double membrane.
Slash
So I
replication.
think
Mhm.
before this we thought that they were double membrane vesicles, but these tubes that link them together, I don't think have been observed before. Um,
Hmm. Oh no, I think again, not maybe not in this case of a coronavirus, but in other versions of this, yes.
okay, I mean that's interesting, and that's quite yeah.
Hmm.
So yeah,
Yeah, so
mm,
basically they're not open to the cytosol, it
mm,
seems, but they're open to each other potentially.
yeah. So
Hmm.
neck-like pretty. So strikingly, thus far unreported tubular connections reaching up to 300 nanometers. And yeah, they're
There's really long
yeah
ones
really
is
long
what they
ones,
haven't seen before.
and
Yeah.
connected to all sorts of different parts of the internal structure of the cells. And yeah, they do demonstrate that with this like 3D
Well,
image.
do they don't say where those other Oh, I see. Between the ER and the
Yes.
DMVs, the endoplasmic reticulum.
So
Hmm.
yeah, and ER is basically the kind of factory of the cell, isn't it? Where you've got like
So much happens
so
in
much
there.
happens in there. Uh, so
I mean, that's where antigen processing is happening, right? So, I mean, that's kind of a surprising thing, right? Like, why would it be connected to the ER? Because there are antigen processing things in the ER. Like, there must be some gating between them, or maybe that's why they're long and tubular because like things don't really diffuse well down there. It's for some other reason.
yeah,
Uh, Other hypotheses. Oh, it could be because this membrane came from the ER, and this is like the remnant of that membrane, right?
yeah,
That like budded off from the ER, and that it's getting smaller and smaller before it becomes like its own center. So like that could be another reason. Then this is sort of the whole purpose of
yeah,
this paper, right? Is like, oh, we see these things like now. how can you explain
Yeah.
it? Because they're there. They use the technique that like doesn't introduce that many artifacts, right? Like electrons are bouncing off of the things that we see, and these things exist inside of the cell. So now, what can explain them? And they don't have explanations for everything.
Yeah.
You just kind of
They,
make
they
them.
have a camera, they're pointing at shooting at stuff, and they're like, okay, well, we'll figure it out later, but finally we've got this great technology. Let's just despite look at as much as we can.
Yeah, yeah, and like right, other scientists will look at this and they'll have their own thoughts and like might inspire their own set of experiments, right, to go down.
Yeah, completely. So I guess does that?
What else do they say? Oh yeah, it's just they're just showing lots of connections in this one. What are the? Do they talk about the black arrows in this one, or is that maybe for later?
Oh, the black arrows. Yeah, they have some black arrows in figure S1H, which is the one right in the middle where they look at.
Mhm.
Uh,
I don't know. They're fusing together.
yeah. Uh, yeah, because I mean the great thing about this paper is like there's like a literal like paragraph describing their observations at the end of each figure, and in the main text they basically go,
Mhm.
oh yeah, we found some stuff. Check the figure's legend. I'm like, okay, fine, and then there's like a ton of like a ton of stuff that would
Yeah,
actually be in the results.
and it's nice,
from
and
that.
they try to tell you about like the frequency in which they see some of these things, right? And like that's again like part of building up that hypothesis. Like, are these things like just are we seeing the ends of the process, the beginning of the process, right? We don't know. It's just a snapshot in time.
Yeah. Okay.
Okay, I
Yeah.
I feel like I've observed all I can out of S1.
Yeah. S3 is the next one.
Yeah, I moved to S3 because
Excellent.
they talk about S2 like closer to the
Oh no,
next
that makes
figure.
sense. Okay, so and S3 is doing some. Let's see what S3 is doing.
So in this one, they're looking specifically at that outer, like you can see how difficult it is to see this stuff.
Yeah.
Looking at the outer membrane of the double membrane vesicle, and they're seeing little dense regions on the outside, electron dense regions, which density here would be black,
Yeah.
right? So things that are electron dense are black.
So
They're absorbing the electrons and not bouncing back into the detector.
yeah, so means there's something like there's some kind of protein or some type of object that electrons are hitting this like not bouncing off of. So
Yep. Yep, not getting through.
yeah,
And here they see stuff. They see stuff. That's all they're
I
saying.
mean
That these aren't smooth membranes with nothing on them. They're full of proteins of some sort.
yeah, I mean yeah. So they basically said that the identity of these different densities could not be ascertained. It is worth noting that numerous viral and host proteins associate with coronavirus replication organelles. So they basically say that okay, we don't know what they are. It could be from the host. They could be from a virus. They could be from aliens. We don't
Yep.
know. We'll find out. But that's yeah,
Yeah, it's kind of interesting. I mean, I wonder whether or not they're even so like what I don't know about this tech is it even possible to get identity on these types of things? Right? Like maybe the resolution they have isn't good enough because like we know that you can't get structural
yes,
information from EM images. So maybe if they had enough images, they could reconstruct what the shape was and then try to match it against other
yeah.
shapes.
That
But that's like a needle, that could be a needle in the haystack because like how many structures do we really know of everything that's possible in the host and the virus, right? We may not know that much.
we may not, but we may be able to make guesses. I mean, we do know structures, and if we have a matching software
That's true.
that's good enough, then
Yeah.
it could potentially be something
Mhm.
in the future.
Yeah.
Uh,
So I mean I think that's like a holy grail of this technology and like sort of big data applied to it, right? Is that like with enough information you could do label-free identification of things. But we're nowhere near that. That's like a pipe dream,
Yeah.
right? Like now if I wanted to know what that is, I would need an antibody raised to that molecule and I would need to attach a little piece of gold to the end of the antibody. And then the next time I image it after it's been soaked with the antibody, I would see, oh yeah, those things they got tagged by this piece of gold.
so next one is figure S4, where we get to see the dsRNA. Apparently, they look focusing
Yes.
more on the inwards on and looking at these filamentous
Mhm.
structures, which I think come out
Mhm.
most clearest in like supplemental C. So
C, yeah, I think C is a good one. You can see how there's, and they're in that one. They're pointing to little dark things. So there are proteins associated, presumably putatively proteins associated with these strands of electron dense material.
yeah,
Probably RNA.
And they do like a 3D molecule of this, what they call filamentous content, that we are assuming is dsRNA. But again, we only have a camera at this point. We can't. There is
Yeah.
the chemistry in this paper. There isn't much of it, but I mean
Yeah.
that's understandable. I mean, because sometimes you have to go out and publish stuff, and from what I've heard, this actual technique takes a lot of labor and takes quite a lot of
Oh, a
yeah,
huge amount of
a huge
time.
amount of time
Yeah.
to actually to do. So, and like I think like only like 60% of these actual slides actually come out, because I think there's quite a lot of ones that break while they're trying to make them, or that kind of bend
Hmm.
so that they don't get good images out of them. So this is
Yep.
so each image there is a lot of blood, sweat, and tears coming out of
Yeah. And we're not even seeing every image that they took, right? There's probably plenty of other good images
yeah.
that were in the store, but didn't have all the features all at once that they wanted to show.
I mean, I get the sense.
So, like, these are probably excerpts from different photos, not just the first tomogram.
Yeah, I really get the sense that they are trying to show us as many images as they can, just to show just to so they get the opportunity to show all of the best images and all the hard work.
Absolutely.
Because usually with
Absolutely.
this, you get one representative image and they go, oh, we looked at a bunch of other images and they also look the same. Whereas here one they're going,
Yep.
no, well, look, I made this. Look at this.
Yes. I mean, it's like it's really good because like that's a criticism that can get levied at microscopy papers, right? Is that you showed us representative, but how do we really know, right, what it is?
Yeah.
And I mean, they didn't give us. So back in the TEM days, I know that people would quantify from TEM fields. They'll say like across this many TEM fields, we saw this many mitochondria or something, right? And like that's how they got at certain numbers even. So they didn't do that level of detail, but they did show us more representative images than one. And they give us a sense. Like, I think in the last one it said like approximately 60% of the things we saw, right, like fell into this category or something like that.
And like even there are like some so tomography can only give you so much, because there's a point where you tilt the image and you can and then the thickness almost doubles, at which point 60%, which point you get very little data. So there's something called like I can't remember. It's still in my mind, but there's like a gap, a shadow, that means that you can only get so much data out of this, which
it's the limitation of tomography.
Yeah, unless we get much thinner, or like someone finds a clever algorithmic way to like subtract different information.
Yeah, different weird. Yeah.
So so yeah, that's let's see.
Yeah, so I mean, most of this is really confirming what we were seeing in one, right? Like there's double membrane vesicles filled with what we think is the viral RNA and covered in proteins, and even inside there's also proteins associating with these strands.
Yep. That brings us to figure S2. Okay. Which is.
Yeah, because now they're focusing in on this membrane and how it's a certain distance apart, 4.5 nanometers. And I guess it's just important. That's like an important measurement because they're going to be focusing on something that bridges that gap, right? So it's it's showing that they can measure these things, right? That they know the space that's going on in here.
Yeah. And the fact that it's such a regular gap kind of is interesting, because that's not necessarily a thing that is set in stone for double membranes. That they could the gaps could be really wide. It's like you have two plastic bags inside of each other, and they don't have to be the fact that there's a regular length is
totally. Yeah. I mean, and that's what they suggest in B, right? They actually see little gray spots that bridge that gap, and they're hypothesizing that that's like a stopper of some sort, right, to keep the distance equivalent.
Yeah.
And that's something we see in bacterial membranes, right? Like in gram negatives, they have those types of proteins.
Yeah, definitely. I mean, especially like around cell division and places like that. I mean, C is particularly interesting because that literally shows the pores holding the membranes together, because there's quite a lot of dissociation there. And I know that they did try to count the number of pores, but again, those numbers, I think there's about 9 pores they managed to establish as their floor. But again, they acknowledge that there's a lot of variation that could be had, because they know they're not seeing everything, and they're very upfront about it.
Yeah, that they haven't done they haven't done enough fields of view to give you a good statistic,
Yeah.
right? Because like fields of view are like this is like such a small percentage of the entire cytoplasm that they're looking at, right? And I think that it's really hard to say because who's to know like where these things get localized? There could be concentrations in different parts of the cell that are different. It's very complicated to make those types of conclusions.
So that brings us to figure 2a. Okay, and this again is looking at the specific double. This like they're focusing more on the pores, right?
Yeah. So 2A is zoomed in, and we're actually seeing like what they think is a pore. They in the inset, that's the zoom in of the pore.
Yeah.
It spans the membrane, and there's sort of like stuff coming
Yeah.
off of it, right? Like you can sort of see like weird spikes at the top, and even at the bottom, there's like an off-center squiggly of some sort.
Yeah.
At bottom, the the inside we call the lumen
Yep.
of these things. So the lumen side and the outer side. Yeah.
And they have SARS-CoV-2 for comparison as well, just to show that this is something that happens in lots of
Yes.
coronaviruses.
Yes. Yes. Yeah. So I mean, relating it back because that's the hot topic these days, right? They're
Yeah.
doing this to tell us about our novel coronavirus friend. What they see in the mirroring one seems to mirror that.
Yeah, definitely. Let's move that on to figure S5, which brings us back to looking at the slices. So from the top
Yep.
end, which let's see what this.
Yeah, so the slices. This is again the arrows. I think in this one are pointing to. I think they're pointing to the. Does it say they're pointing to the pores? Yeah. The arrows are pointing to the pores here. I mean, it's hard to see maybe in this zoomed out one, so we'll take their word for it, or you can look at the paper yourself and zoom in at each arrowhead. But the point again is similar to what we just saw, that in these two slices, one from the murine virus and one from our novel coronavirus, the same structure is there. That pore exists between the two.
Yeah.
Yeah, and I think what I like also here is that we're really seeing that zoomed out image A and C of the slice.
Yeah.
And you can see they're not like, it's not perfect, right? Like they have sections that got blasted in different ways, maybe some holes, some things got flaky and they crumbled away. Actually, that's actually really evident in C. At the very top, there's those missing parts, right? And like those are probably like lipid droplets or organelles that like fell out of the slice.
Yeah, of course. Yeah.
Right? Because like a really thin slice, like what are the forces that are holding everything together? It's just on that really paper thin thing. So like something really dense and heavy would just like drop out sometimes.
Yeah.
So anyways, that's just you can see how many things can go wrong with getting a nice image.
Yeah. And then figure 2C and M. So I'll start off with C because I think I did make a quick GIF for figure 2D but
oh nice.
But it focuses in on that pore and looking at those weird structures around it.
Yeah, and I think this is an averaged view now. So after seeing many of them, right, after seeing many pores in different orientations, right, the computer compiles that into what is the average of all of that. And it's really a cloud, right? It's a big cloud, a 3D structure cloud. And then this is the slice, the middle slice of that cloud.
Oh, it's like those average of human faces that people do so to make everyone look like
yes,
clowns, like
it
that,
is.
but
It is
for
just
protein.
like it is just like yes, and hopefully proteins have less variation than human faces.
Hopefully.
And the variation that you see would be places of motion, right? Is what it is.
Yeah.
So like stuff that's really dark is always there. It's like that's probably like the rigid parts of the protein, or even like you see the double membrane here, right?
Yeah.
It could be we don't know what it is, electron dense stuff. And so like you can see it's rigid, right? Like there's nothing in between the membranes, right? That's white. We never really see anything there. But outside in the lumen and the outer membrane, there's all sorts of stuff sometimes. It's floating around. So it's like a light gray. And as you get closer, you can even see other density areas. And I think we'll discuss it more like later in the paper.
And based off that, they've built this model of how how it works.
Yeah.
By looking at almost like building up a top-down image and then scaling it down at different points.
Mm-hmm.
So
Yeah, well, I mean, again, this is the cloud that they have, and you can slice and dice it different ways. A really nice way. I like these 3D images. What it doesn't communicate to you is that uncertainty,
Yeah,
right? So in the gray, you can get a sense that things that are light gray are more uncertain, and things that are dark, like they aren't as much. Oh, I love this GIF. This is really great.
yeah,
So yeah, you can but easy for the human brain to understand is like a 3D model. So
yeah,
they choose like a cutoff point of like darkness, and they say that stuff is
yeah,
there, and then they fill it in for us so we can see what that looks like.
yeah.
But of course, like those slices that we're going through right now, like that is the raw data that's come out. Well, it's not even raw. It's the process. It's one of the processing steps where they saw so many different things and they average it all together.
Yeah, and you can see the areas where there are like gaps and like, and the way it differs when they're looking at stuff that's in the membrane, because of course there's this comparison going on as well.
Yeah, so like slice J or something, there's like a lot of density
Yeah,
that some of that density might be membrane, right? And like the way that it kind of J and K, the way that it curves off, there's a curvature to this whole thing,
yeah,
right? So that it's also sort of it can even show us that in these images.
yeah. Okay, so oh yeah, I can do the next movie as well. Let's see if I can set that up quickly. Um, let's see. So the next movie
Yeah, so
is
oh, you're
is
going.
go. Sorry, no, you go ahead. You can explain.
Oh, I was just gonna say, like, from this tomogram, subtomogram, right? Because it's just they're focusing on this one thing, average version. You really get a sense of the shape, right? Like that on the outer side, there are these flanges, right? On the luminal side, there's like nothing much. We'll talk a little bit more about that. And then there's a channel that goes all the way through, and they can measure all those distances, right? 14 nanometers from the center to the edge. It's about 13 nanometers long. Tiny, tiny things to be looking at.
Yeah. Um,
So this is the GIF, or this is the movie that they gave us. It's the same as your GIF, kind of, right?
yeah,
That it's smoothly moving through the sections from outer side to luminal side. No, from lumen to outside. Yeah, yeah. It goes out.
yeah. Um, and then
Coming
after
back in now.
the movie, you've got yeah, we've got lots of. We've got so our budget has gone so much higher now. We've got so many animations. We've got. I mean, we're making the big leagues. So yeah, figure 6A and D, where these are ones where I didn't really understand these as much. Um,
Yeah, so I mean, this is right because as I said, or as I was saying before, B is a good one to look at here,
yeah,
right? This is an average, right? Like the stuff that's in light gray is stuff that they're more uncertain about, and so you can actually assign like a resolution number to each of those points.
Right.
Like the resolution of the image isn't perfect because the places where things move around are much or that are shadowed, right? Shadow also means that the resolution is who knows what it is in there. That drives this. So I was actually reading this about like the difference between EM structures and crystallography structures for proteins. In that in crystallography structures, you usually have the value of like the resolution of it sort of applies to the whole thing because the crystal is a regularly repeating structure. You get the same kind of image from every point, but here it's not the same because like we don't control the orientation of these molecules as we see them in their frozen state, right? We may see more top down than we do side, right? We have no idea. It's all over the place. And so the data means that the model itself has uncertainties in the areas. But I mean, you can see it's pretty good,
Yeah.
right? Like the uncertainty is in that middle chunk, which actually, as we'll like talk about later, might not even be part of the protein, right? That could be the uncertainty of like something that is flexible in that region, maybe something moving through the pore.
Yeah, exactly. It's like if you're doing like what's it, a long exposure image of like traffic, and suddenly you see this like there's the road's there, and you see this smear that's actually all these cars going through. That they're not part of the motorway, but they register on that image because there's always something going through, and so
Right.
it's almost like that where we've got this long exposure of how this protein is acting, and there are these parts that are just we're not sure how they're working.
Yeah. And then like A and C, I think are more into the maths of this. And again, I'm not super familiar with it, but you have to imagine that like when they made these 3D models are made from all these different orientations, right? And they've averaged them together in some way. And you can make assumptions, right, about the molecule that might improve the resolution in some, right, improve what you're seeing out there in the raw data, right, in that cloud of all those different orientations. And what they're doing here, the C1, it's wild type C6 in A and wild type C1. That is like the assumption of six-fold symmetry. Right. And that's because like we know pores. We've seen pores in different circumstances, right? And a human that looked at these images said, well, you know, this pore looks like it might have six-fold symmetry. Well, for a computer, that's the world of a difference, right? Because for a computer, that's an assumption that they don't have to change parameters with, right? And play around within the density. So for someone to make that decision and make the assumption that there's six-fold symmetry in this molecule, it constrains the algorithm and lets you get a better resolution.
Yeah.
Yeah. So that's what A. So
Okay.
A is sort of showing us that, I believe, and then I think similarly C is another way of showing it.
Yeah.
Basically, to show that they made an assumption to get a good to improve their resolution.
Yeah.
And that assumption is warranted because we've seen lots of these molecules before, because it gives us better correlation with the data. So like it explains more of what we're seeing to say that there's six-fold symmetry here.
Yeah, that's great. I mean, explanatory power is important. I want to also GFP C6. Do you want to talk a little bit about that?
Yeah.
Yeah. Or
no. Let's move on to the next one because that's part of their next experimental setup.
Okay. So we've got figure 6 S6E to F, which again this I mean it gives us almost the same information as those fancy videos, but you get to see each
exactly just each individual one. So actually, we never saw the information in the X slides, right? C1 versus C6, or it's all C6 actually. Yes. Really is the one we should be focused on. But we've never seen it like pass through in that direction, all the slices. And so they're just showing us the raw data here, or not the raw, but the process tomogram in all the different slices. And I guess maybe I don't know if you can see any difference between C1 and C6, but the idea is that C6 is supposed to have more certainty to it. Right. Because when they made that assumption, then more stuff like came out as like, this is the part of the protein that we know exists. Yeah. Just more images, more background to show that they did the work.
Yeah. And then figure 3A, where you're looking at about the coding. So they don't know what the pore is, but they're trying to predict what's in it by looking at coding sequences.
Yeah. So they want to know. And actually, I guess that we didn't really touch on that last figure in 6AD. But 6AD, like because they know the shape and the general volume of things from these tomographic images, they can make assumptions on how heavy everything is, right? Because you know how much space it takes up. You know, like the density of a protein, and so you come up with some estimation. And from that, we know the coding sequences of everything in the coronavirus genome, which gives us sequence structure that also gives us estimates, or actually gives us a more precise weight for everything. And from all of that, they think that it might be this NSP3, I think. Is that what they think it is? Yes.
Well, let me double check this because the thing is, what was NSP?
Delta 2 GFP. Yeah, it's NSP3. They think that NSP3 is part of the pore, at least.
Yes, that's right.
Right? They're not sure all the parts, but because of the size of NSP3 and the fact that it has these transmembrane domains, right? That they have regions on both sides. That's a good candidate for something that embeds into a double membrane and that is on both sides.
And I think how they test it, like how they confirm that it's in there, is quite interesting.
So now they want to know, and so they below you can see they have the construct they're going to test. They have a delta 2 GFP NSP3.
Yeah. So I mean, the interesting thing is viruses can only handle so many like kind of kilobytes of information inside them. They can only handle a certain length of DNA. So if you want to add in something, you have to take something out. So they had to take out a gene that hopefully doesn't do anything, or that doesn't like mess up the That doesn't stop the
Yeah,
virus from replicating if they remove it.
because they still need the gene to appear, like to make they still have to make these organelles.
Yeah.
The virus can't be so deficient it can't get to this stage.
So yeah, they took out one bit of that gene and put in this EGFP to look for seeing whether that's encoded in the pores.
I think also for correlative to make sure that like they actually because you're infecting a whole bunch of cells, not every cell might get infected, right? I think they also want the EGFP as a marker to say like, oh, this is where this is where it is.
Yeah.
So wait, delta 2. What does the delta 2 mean in this sense? I forget.
One NSP2, which is apparently dispensable for replication in cell culture. So
I see. They remove one of the proteins, and then they also tag another one.
Yeah. So in order, they had to get rid of NSP2 to include this GFP protein in there. And fortunately, NSP2 doesn't stop the virus from replicating in cell culture. So hopefully that means that all the replication kind of steps should still happen.
Right.
So
right.
But obviously there is always that worry that will it make a difference? So they do take some images to try and confirm that for this specific aspect of the disease it does correlate.
Mm-hmm.
So
yeah, let's move on to B of this figure then, or I guess S7 is
S7 where we get to see that GFP in action, where they shine it onto cells and you can see all these cells expressing GFP, and
so like this is I also think this is a wild element of the technology, right? Is that they're able to correlate what they're doing in EM with fluorescence. Yeah. And there's so many more tools in fluorescence for marking things. And yeah, so here, as I said, they don't have to worry about finding that the cell that they're targeting for this technique isn't infected because they know it's infected because it has GFP.
Yeah, it saves them a lot of time, and I think that's because with these sorts of things you can spend years looking through every cell to try and find one that's infected. So that sort of time saving actually is really, really important for when you're working as a scientist.
So they're able to find that cell. You can see again they have the C and D, the before and after. They blow away a section with ion fire.
Yeah, ion beams. We are the future.
Ion beam fire. And they show you C and E where they actually look down and then they zoom in to F. And they got them. They got pores.
Yeah, they got pores, so it works.
It works. And zooming into the pores, I guess, is the next.
Yes.
Is our next.
So next we're going to be looking at figure S6F, which again, like the previous figure when you're looking at ENF, C6, but comparing it with the GFP version to see that, and we can actually see that the difference isn't that big.
Not looking at it like this, at least, right? It's hard to see what's going on. I definitely think.
I'm looking at a magic eye picture and kind of crossing my eyes and trying to see where they fit together.
I see, for me, in the second row, near the end, the last 3 images, they look pretty different between F and G. Something's changed there.
Yeah.
Yeah. There's more Y action happening on the F side.
Yeah, that's true.
But anyways, this is again, they went through this process. They were able to get this, but really, if we saw just a single slice, it's probably easier for humans.
But yeah, the fact that something is going slightly wrong with it does indicate that this is to do with the coronavirus, because if it's having no effect, then that might be a signal that this might be a cell-related process. But the fact that if you mess up the coronavirus slightly and this changes, you go, that they're related. Oh,
sure. Yeah. I think this is a really good, I agree. This is a great, confirmation that, these pores aren't coming from the host.
Yeah.
Right? They're likely not coming from the host.
Yeah, this is equivalent of punching the coronavirus. If it goes, ow, then when you've hit something that's...
Yeah, exactly. Which, I mean, it's a very classic genetic technique. Punch the genome and see what happens.
That's literally how I'm going to teach gene. Yeah. So figure 3BD, which again looks more in depth at the pore, and it also looks.
Yeah. So this is something that's easier to see, right? Between C and D, we can see the difference between the wild type and the GFP. And they helped us. They even highlighted it with this difference map.
Yes. Yeah, and you can see the GFP almost looks a bit stiffer. There's like a lot more density in certain areas, whereas the wild type there's a lot more flexibility there.
Well, I mean, I think presumably in the difference map it means that that might be the
Oh
GFP,
yeah, no,
right?
you're right, of course, because GFP is like a big barrel protein that again adds more
Yeah.
density. So
Yeah.
yeah,
So they like stuck that GFP at the end. You know, what's interesting is that there's also difference in the channel itself, in the pore itself.
yes,
Uh, so like I guess, you know, like we don't really know how the pore gets formed, right? Like how do these proteins get, you know, they don't just magically insert themselves. There's machinery potentially that inserts them. Or the protein itself has evolved, right? To as it's inserting itself form the channel and the symmetry. I don't know, there's so many unknowns
yeah,
in this realm. But it's interesting to see that just the addition of this chunk like can change that. I mean, it could even be NSP. You know, I mean, you can't rule it out.
yeah.
NSP2 could be doing something here, right? Like, what if NSP2 is part of the chaperone system that like helps these things insert? Again, I have no idea. I don't know what
Yeah,
NSP2
this is. I mean,
is like.
we're about asking questions here because this is like we bring presented the images, and scientists can. Because yeah, there's a lot of interpretation that's put into it, but we don't necessarily have to take every. We have to. More interpretation will come as more data will appear, and it'll either
Absolutely.
confirm what the researchers have suggested, which is potentially likely, or it might be something completely insane, which is probably very unlikely, but very fun to think about.
Yeah, yeah, yeah. I don't know if they said. I think they did say that they thought it was EGFP that's out there.
Yeah, I mean that would make sense. I mean,
Figure 3. Oh, let's see the text here.
yeah.
Oh yeah, well in E, no, in E they say revealed the presence of additional masses, and they put in brackets EGFP. So they do think that that's
Yeah,
EGFP.
and that would make
But
sense.
but it, yeah, but again, but it doesn't explain the density difference in the pore channel center, right? That to me, that's like maybe a badly formed pore.
Yeah.
But yeah, don't know there.
It could easily be that the way it's adding GFP at the N terminal means that the entire protein is changed or like less able to function. So certain parts will be more static.
Yep, absolutely.
But yeah, it's for protein science for me.
Oh, actually, I one thing that I'm noticing now when I'm looking at this is there's slight green on the inner face of both the luminal and the outer membrane.
Yeah.
Right, there's like these slight green dots there. I want that. I could bring a hypothesis to that one. It could be curvature changes on the membrane.
Ooh. Oh,
right, because like we know that like proteins, like when they engage the membrane, they're helping to form, they can help form that curvature in some ways, right? We even in this paper they had examples of the spacers, right? And stuff that changed the distance. So like this mutation adding that GFP also like because the pore didn't form properly, also maybe pulled the membrane out of what we saw before orientation. Maybe. I don't know. You know, just throwing stuff out there.
No, I mean, we're throwing stuff out there. We're reading papers and we're trying to like get into the data and yeah, get creative a little bit sometimes. And sometimes it's quite fun to ask these questions because usually what happens is sometimes people ask these questions and go, you know what? I'll do an experiment and sometimes that can turn out to be quite an interesting avenue, or you can go into the file drawer and no one will see it. Like, oh yeah. So where did your grant money go? Well, I tried to do something, but it didn't work. Oh.
Yeah. Well, I mean, I hopefully like in the future we'll just publish all those things in the folder as well, right? Right now it's like some system of competition that things get hidden like that.
I'm hoping for that. But then again, I run an open access journal that is like all about like publish all your data for us for a reasonable price and. So, obviously my biases are very clear in that direction, but you know, you can always stick your data in the file drawer and no one has to see it if you. But I mean, we're both open access advocates, I believe. So, yeah, we want to see all your dirty data, even if it looks terrible. We want to see it so we can learn from it.
All right, let's move on. Let's see what else we got.
The next movie. So I can transition to that. So
yeah, but this movie is pretty simple actually. It's I think 2 images flickering back and forth, just so that we can see that difference map a little bit easier. If you don't believe them.
Yeah, no, it's like going to the optician is better. worse, better, worse.
Yeah, exactly.
But yeah, no, that definitely makes the point a bit more apparent. Move over to the next one, figure S8, and I've gone cross-eyed. This, yeah, there's a lot of images there.
Yeah. More pores.
More pores.
Right. More pores. Yeah, they show us a lot of different things. I think now they're really trying to look at maybe things that could associate with the pores,
Yeah.
right? They're kind of pointing out like, oh, look, sometimes there's like a little bit extra out here. And like in the subtomogram, that's going to just show up as like light gray, like uncertainty, right? Like we don't know because it's not in every picture. But when you take a human look to it, you're like, oh yeah, well, sometimes like one side looks longer than the other, and sometimes there's like strand-like things and coming out of the bottom. I think that's what they're also
chain-like
pointing here.
Associating with the complex prongs, and large assemblies in some cases are reminiscent of helical coronavirus RNTs, so ribonuclease proteins. So
Yeah. So as the genome is being pumped out or as it's going out, like it's also getting covered, right, with protein. And that's really important because you don't want that free RNA just floating around in the cell. Someone's going to notice it. So again, like this is part of that building that hypothesis, the story really, that the reason why these pores, or the reason why these double membrane vesicles exist is to protect them from the cytosol, and that the pores are the way of getting the RNA you need out, and that means they have to associate with all those other factors that are helping to make sure that RNA is getting protected, that it's being translated properly out in the cytoplasm. Yeah, they don't say that they see ribosomes inside of the vesicles, right? They do see punctate things, but they never call them ribosomes, I don't think. So, I mean, that's an interesting question. Do ribosomes get into these replication vesicles? Because if they don't, then you're stuck with whatever protein gets in there, right? At the beginning, you don't make more. And again, that's all part of understanding how does this virus replicate, like real mechanistically, right? We know it gets in and it gets out, but all the steps along the way, this is fleshing out that story.
Yeah, exactly. So that figure S8 does a lot of that, and figure 2CM is the next one. So going back to the coolest figure, but then we're looking at the underside.
Oh yeah. Yeah, because it kind of links to what they were saying in 8, right? In 8, they started talking about, oh, like these pores, maybe like they want to say that it does export out the RNA. And so even in the tomogram, they see that at the bottom of it, there's something there.
Yeah.
And it's off, and they say it's off center, right? It's not part of the symmetry, I think is something important that they point out. So it might not even be RNA they're picking up. I think they also hypothesize there's something that sits there, like a protein of some sort that helps do the pushing.
Like maybe like it gets associated with some like RDRP, or maybe like something like that. Without the protein, or maybe if the RNA is going through and the RDRP is still like latched on the other end, it's like kind of bumps up and down against it or something.
Right. Yeah. So it could be like a ratchet. There's all these things that happen. We don't know how does this pore. We assume the pore is for export of RNA. How does it do that? Again, here's like another clue maybe that they're throwing our way, right? That they see this. And in M, you can see that like how it's off center, right? Like C1. It's like, there is some sort of mass there that hangs out near the bottom.
I mean, in M, they like color it green, so it looks like a bit of snot that's dangling out the bottom of the of the pore.
Oh, excuse me,
I've had those days.
Pore has
pores as well.
On the luminal side.
Yeah.
That's all I want to say here.
Yeah, no, that's important, and that builds into that information that this is like a complex that's doing something, that's transporting something. We don't know what yet, but there's something there.
Yeah. And it's not doing it by itself, right? It's not like it's a self-contained machine. I mean, yes, it's a machine of some sort that has all these parts, but there's other stuff interfacing with it potentially, other proteins. Like, for example, when people see things that have like collared rims like that, they're often thought of as association, like assembly points, right? Where other proteins latch on, and that's where you start getting more complicated machinery to, like, I don't know, do something.
Yeah. Figure 9 is like where you're actually seeing virus particles being assembled inside.
Yeah.
So this is quite a neat, a nice surprise.
Yeah. So So this is not in these are not on the DMVs themselves, right? I think
Yeah.
these are just other sections where they are seeing, viral particles being created. So that I guess they want to say that it happens in close proximity to each other,
Yeah.
right? Like these DMVs and viral assembly, like they're not, in distant parts of the cell. They're happening in an area, a section.
Yeah.
Which I just love this view of biology. First of all, it obviously it happened. We know in biology there's going to be like local regions, but it just makes everything so much more complicated because the biochemical view is it happens in the cell. But like beyond that, you don't know.
Yeah.
And then here it's like there's a region in the cells where we're seeing DMVs and viral replication. Like it's this zone that's getting hijacked.
Yeah. I mean, I think I've been finding that ever like in like school. I was taught like, oh yeah, cells. There's like big like sacks of stuff that mix together, and then the more we like
Right.
learn about them, the more we realize that there's so much order in there. There's like
Highly structured.
structured.
Yeah, even when it doesn't look structured, it is structured. Like
Yeah.
yeah.
Like viruses aren't alive yet we are seeing them making their own cell organelles that are specific to viruses, and then we've got them like assembling each other and like almost doing all these biological things inside the cell that you wouldn't really suspect them of. Because the usual story is like,
Yeah.
oh, the virus puts their DNA and our silly like machinery just like puts it together for them. But this actually
Exactly.
shows the virus is taking a much more active role.
Yeah, there's way more. I mean, like just knowing that there's all these intracellular like immune monitoring things, right? Like cells aren't that naive to just take any old sequence and make it. Yeah, stuff's going on.
Yeah.
Yeah, they even point out the spikes, right? So in
Yeah.
C, you can see that's the classic coronavirus.
Yeah.
Hello coronavirus with the tiny corona that it has, because of those spike proteins. It's kind of nice to see it. At one point I wanted to flash back and forth between like that image that they're showing on the news all the time, right? Because that's it. That's our
Yeah.
coronavirus. Well, this is the mouse hepatitis version, but where the namesake comes from.
But you really won't be able to tell the difference. They're very difficult. I
You
think.
really, yeah, you cannot tell at the EM level yet. We don't have the resolution yet.
Not yet.
Someday. Someday.
And that brings us to the final figure. Figure 4, they pull all the story together, and yeah, so they show like essentially their model of the coronavirus replication and what it's doing inside the cell based on these images.
Yes. Yeah. So they have the EM image where they think the thing is happening, and they gave us a nice cartoon to show us like what's going on in that sense as well. Yeah, and that's sort of what we've been talking about this whole time. Replication is happening, genome replication and subgenomic RNA replication is happening inside. Actually, they don't even really talk to that. I think they talk about genome replication, right? But what about the subgene? What about the stuff that makes the accessory proteins? Anyways,
I mean, we don't know.
That's not in the story.
I mean, maybe that's happening inside the replication organelle. Maybe that's why it's connected to the ER. But we don't know. That's
Oh wow, that'd be kind of interesting.
That's massive speculation on my part. If it turns out to be true, I'll kick myself. But I'll be like, damn, I could have gone there first. But
yeah, no idea. I mean, when I asked before, or when I was thinking before, they never really talk about ribosomes in these double membrane vesicles, right? Like that's another thing. Like if there's no ribosomes, then it doesn't happen inside. The subgenomic would have to go outside. But maybe it's easier to have subgenomics flying around on the outside because that's just like messenger RNA, right? In the inside, that's like double-stranded because like they're making both versions, and eventually they'll have to make like the full genomics. So I don't know.
They need the RDRP to be in there, the ribosome, the RNA-dependent polymerases to be in there, and the question is, how do those get in there? Where did they come from?
How did they get in there?
That's
what. Absolutely.
And yeah, this is so I think, and this has been quite a good.
I was wondering. I don't even know. Do they can't differentiate between like an uncoiling polypeptide, right? That's going in versus coming out.
Yeah.
I mean, this is maybe there's no precedent for this. Like one of the reasons why they're talking about this pore as if it exports RNA is because pores like this exist in other RNA viruses. Right. I think in the introduction they talked about reovirus, which is sort of the classical double-stranded RNA genome virus, and it doesn't make these vesicles, but it has a capsid, and the capsid has a pore that shoots stuff out of it. So like that's why we're thinking of the pore this way. But I wonder what prevents them from thinking about the pore as exporting, like being able to import protein.
Oh my god,
Right? What if
that's trippy. That's it's like a vacuum cleaner. It's like sucking up all the viral RNA. I,
Yeah, well, no, not the viral RNA. What if it's importing? What if it can import protein?
oh,
So
right.
that, like, to get you asked, how does the RNA dependent RNA polymerase get in? Like, what if one of the other stage, like with a certain association on the outside of the pore, it actually sucks things in. Yeah, but I mean, even at the sense of sucking in viral RNA, like that's like a nucleus style
Yeah.
almost type thing, right? Like nucleus do that.
Yeah.
Nucleus suck stuff and export things from their pore.
But
Anyways,
yeah.
though that their pore is also way more complicated than this pore. Their pore is made of multiple
But
proteins. This is
yeah,
just a
I mean,
single protein.
this is the kind of paper where it does allow that space for speculating on this kind of way, and it allows us to
Yeah.
look and kind of because we're seeing these images that no one's really captured before because you've got this whole new revolution, and it's like
Yeah.
yeah, our imagines can run wild. I'm like, oh look, there are canals on Mars. That's obvious. That's the kind of thing that like, oh look, there's yeah,
It's exactly that. I think that that's a great comparison, actually, right? Like seeing these things is like seeing structures on planets, or like that. Well, what was that encapsulated like ice thing, right? That people were like, that could be a spaceship.
oh god. I mean, I'm thinking about the face on Mars, or like the very
Oh, the face on Mars. Yeah,
the
that's a good one
the
too.
smiley face, and then there's like that. Yeah, there's all sorts of. But the
Yeah.
canals. I mean, oh yeah, they could be.
I mean,
Sorry,
it's only a matter of time before someone sees the face inside of a cell, right?
I'm sure that must have happened already. I'm 90% sure. I feel like I've already seen that before, and like in this, my slides go, yeah, there's a smiley face,
Yeah.
and then I just move on. But I mean, especially because I was watching streptococci, so they naturally form into smiley faces sometimes. So it's
Oh yeah. They were communicating with you. Did you know, Buzz? They were trying to say, happy to be here.
yeah, they're like, ah, yeah, that isn't that great. But yeah, one of the things that I found interesting about this is that what does this mean for drugs? Because there's a lot of drugs that are targeting the RDRP like complex, and so if that's in these double membrane vesicles, then that raises questions of
Oh
like
yeah.
how
Amazing. Yeah, that is a really good thought. Like, yeah, not only does the drug have to be diffused through the cell membrane, but it has to be able to diffuse through this membrane.
yeah. So like the
Oh, and if it's being delivered, sorry,
yeah,
I
go for
just
it.
jumped on. If it's being delivered in a way, like if it's like a lipid caps encapsulated, right? I know that there's like some formulations that they like mix up the drug in lipid molecules so that it can get in and like fuse to the membrane. Then it would also be less effective because once you fused once and got into the cytosol, you can't get through the second
Yeah.
membrane.
So I mean, I mean, because I know like remdesivir has a portion on it that helps deliver that kind of the main like nucleotide analog dissociates with,
Yep.
and so
Mm-hmm.
could that be an interfere if like that does if that could this interfere with that process and also thinking of
Yeah.
targeting this in the future like I mean targeting this pore so having a nucleoside that can go backwards through this pore if this is taking stuff up then having something that can do that might make a more of
Yeah.
an antiviral.
For sure. Or just something that could plug it, right? Like also the pore, there's not really, as I said before, like the nuclear pore is not like this. It's made of multiple
Yeah.
proteins. This protein is pretty unique, I
Yeah.
think. Might be very unique to the state of actually, don't RNAs go into the endoplasmic reticulum, I think? They can, right?
Yeah.
Because like the rough endoplasmic reticulum, it's covered with
Yeah.
ribosomes where oh no, they're injecting the protein into they're not injecting the RNA.
Yeah.
Anyways, I was just thinking about other places. So I don't think there's many places in the cell where RNA passes through a pore. So yeah, you're right. That's a very unique target for some sort of blocker, small molecule blocker.
Yeah. Yeah, I think this is so. So I think that's been all the figures for that paper, right? Yeah.
Yeah. I mean, that's sort of all. I feel like that's the discussion too. Of that's like those are really good like application-minded thoughts, right? That have come out from this, and throughout we've been talking about all of the open questions of the actual viral biology that need to be followed up with. But yeah, I like this paper. It's kind of light in some ways because it is just images.
Yeah.
So like really, it's like people's knowledge of the literature, I guess, is going to help inform like the analysis of all this stuff. But otherwise, you can just kind of go off on your own tangent in terms of like talking about what you might be possible because they have no identity to any of this stuff.
There's a bit of like questions about like, oh, there's a lot of algorithms they use to try and get these, and the physics of this that's simple. Because I mean, microscopy is quite a lot of physics, and we're not necessarily equipped to deal with that. So there might be some like artifacts that a physicist might see that we won't. But
yes. Yeah, like if we're saying like, oh, like they say, oh, that's GFP, right? When they added it, it means they tagged it. But what if that's a artifact that could be produced in some way, right? Like how can they confirm that sort of
GFP,
thing?
it brings in like a metal ion, I think, inside of it and that could affect the way that it appears on electron microscopy. So we don't.
Yeah.
And
we can't bring that type of criticism, right? Because we're not experts in that way. Like that's not the type of thing we bring when we read this paper.
If you're in the audience and
but what we can bring,
if there are physicists watching this in the audience and you made it this far, thank you, and you can tweet like responses and point out when we're being crazy and making assumptions like, okay, we found a pyramid on Mars in this cell picture. Like,
absolutely, yeah, and share your favorite hypotheses, right, of like what you think might be happening. Because I think that's also part of this, right? It's the data for us to look at.
Yeah, the great thing about open access is that everyone can discuss, and the data's out, and we can look at the shortcomings. And yeah, especially with this, we can be a bit more creative because at least with this, unlike with treatment papers, people's like health isn't online when we're looking at how this, not directly. So
yeah, like our interpretation of this hopefully isn't pushing like some public discourse, right, about about treatment options.
Yeah, so I feel a lot more free to talk about that than if we were talking about like a vaccine paper. Then I'd be much more careful about my speculations.
Absolutely.
But yeah.
So if you guys are interested in having us discuss a paper, or you have other questions with this paper, reach out to us on Twitter. Next week we'll come together and we'll talk about some news headlines and try to choose the paper for next week.
Yeah, thank you so much for joining me. I'll see you next week.
All right.
All right, bye.